Hey, good morning, everybody, or good afternoon, or good evening, wherever you are in the world. It's great to have you here on the International Day for Biological Diversity 2024. Thanks for joining us to learn all about aquatic fungi diversity. I'm Moni Boehm. I'm the Freshwater Conservation Coordinator at the Global Center for Species Survival at the Indianapolis Zoo. The Global Center for Species Survival, just a very brief introduction, is a partnership between the Indianapolis Zoo and the International Union for the Conservation of Nature's Species Survival Commission. Essentially, our job is to support the various specialist groups and all the fantastic work that they do to protect species around the world. We have a team of folks, as you can see. We really are focusing on all the different critters and plants and fungi out there. And part of this, of course, is to support the IUCN Aquatic Fungi Specialist Group. I'll hand over now to some of my co-hosts to introduce their projects. Jenny?
Hi, everyone. My name is Jenny Anderson, and it's my privilege to be a partner and the coordinator of the project FUNACTION, where we are working towards the conservation of aquatic fungi. We have three bases to our work. We are focused on producing the foundational knowledge that we need to begin the conservation of aquatic fungi. We're committed to translating that knowledge into conservation planning, and we're working very hard to communicate about aquatic fungi. So that's part of our mission today, is informing and introducing these wonderful organisms to more people in the world. If you can change the next slide, please. Thank you. So we're a very large group now, working across Europe and the US. And I could talk for hours about this group, but I would just invite you to join us at FUNACTION EU, or online at X, or on LinkedIn to follow our activities and to learn more about us.
Hello, thanks for joining. I'm Andy Bru, the coordinator of MoSTFun, and also of an amazing and diverse team based mainly in Europe and North America. MoSTFun has started about a month ago and will, over the next 3 years, develop the strategies and approaches and tools needed for routine monitoring of aquatic fungi biodiversity. If you can move the slide, please, Moni. Thank you. Yeah, currently aquatic fungi are not monitored, which under the current scenarios of biodiversity loss is also a risk for the important ecosystem processes and services that they are providing for the ecosystems and for human societies. We will achieve the goals of the project by reanalyzing data and samples, but also approaches and experiences from existing biodiversity monitoring programs. These projects, however, were not designed for aquatic fungi. They were designed for other organism groups. Nevertheless, we will ensure the uptake of our projects through a strong stakeholder engagement program, and also by using existing biodiversity monitoring concepts. So these products can be uptaken by stakeholders and other end user groups, and supports the establishment of biodiversity monitoring programs for aquatic fungi biodiversity in Europe, but also in other regions of the world. Thank you.
Hello, everyone. I'm here representing the newly created IUCN Species Survival Commission Aquatic Fungi Specialist Group. It is my pleasure to be here representing the group and also co-organizing this webinar today to celebrate aquatic fungi diversity. This group was first, let's say, the idea first developed on the consortium of FUNACTION project, but then several other colleagues from around the world, both experts in freshwater and marine fungi, joined us to form this group that was created end of last year. And our vision is to develop the scientific and coordination basis for the first conservation assessment, planning, and action efforts for aquatic fungi, both freshwater and marine. There will be several other activities coming coming on in the next few weeks and months. So follow us to get updates on our activities. And that's it on our side for now. We are here today to celebrate aquatic fungi diversity, and that's what we are going to do. And with that, I start introducing our first speaker. Just before I do that, just to remember everyone that there will be a question and answers time. So please make your questions on the Q&A section. And on to present our first speaker today, which is Laura Garzoli. So Laura, she did her PhD on marine fungi and their application in biotechnology. She worked for several years as a taxonomist and also as a curator of the marine culture collection at University of Turin in Italy, where she described 2 new species of marine ascomycetes. She is now a researcher at the National Research Council, Water Research Institute of Verbania, Italy, where she is the head of the Aquatic Microbial Culture Collection, MEGIC. I hope I have said it correctly. She's the PI of the Italian partner of FUNACTION and MoSTFun projects, and she's also responsible for the CNR of the project SUS MIRI IT. Laura, I hand a lot to you. Now you can present, you can start your presentation. Thank you so much.
Thank you, Isabel, for this nice presentation, very flattering. And hi, everyone. Welcome to our Biodiversity Day 2024. I will share my screen now. So, I'm Laura Garzoli, and today I want to take you to have a tour on the hidden realm of marine fungi. I'm very happy to be here on the behalf of FUNACTION and MoSTFun projects, which I'm part of. If you want to be a real expert in marine fungi, then I thought that it would be nice to answer these 5W questions. So who, what, where, and when, and why. So first question, to be a real expert about marine fungi is what? This is the reaction I got when I talk to my relatives and friends when I say I'm working on the marine fungi. So what they are these organisms? They are mushrooms that grow under the sea. What they are? Well, let's start with another question then. So these are all the shapes that you can find under the sea. Really beautiful fungi that are able to float and swim. But also, if you are a mycologist, you can recognize some species that you can find also in terrestrial environment. When does it start, the study of marine fungi? Well, in the late 19th centuries, there was the first 2 description of species from marine phycologists. So this scientist that studies marine plants described the fungi associated to these marine plants. But the real success of marine mycologists started later at the half of the 20th centuries by these 2 publications, the one from 1944, Marine Mycology, Their Taxonomy and Biology, and later in 1979. But this is like the key textbook to have if you want to study marine mycology is by Kohlmeier and Kohlmeier that describe all these marine fungi. And then, of course, there was the advent of molecular biology. So this diversity and study increase. And today, also with the advent of metagenomic and high throughput sequencing, we are started to know that marine fungal diversity is really important in the marine environment, and many more studies are made today about their diversity and role. But who they are? Kohlmeier and Kohlmeier describe them into groups. Obligate marine fungi that you can see here. This is one species, Corolospora maritima, are those that grows and sporulate exclusively in marine or estuarine habitats. This species has a floating appendage And facultative marine fungi are those that are maybe from freshwater or terrestrial environments, but they are able to survive and reproduce in the marine environment. So you can see here is a strain of Scolecobasidium arenarium that you can find in terrestrial environments, but it can grow better at the concentration of sea salt the same as in the marine environment. But later this concept evolved, and now marine fungi are considered an ecological rather than a taxonomical group. They are defined as any fungi that is recovered repeatedly from marine habitats. They are able to grow or sporulate in marine environments. They are able to form symbiotic relationships, or they can adapt, evolve, or they are just active metabolically in the marine environment. You can see here that almost all phylum of the kingdom fungi is represented in the marine environment. And interestingly, with high throughput sequencing, some groups, some phyla, like chytridiomycetes, that have been before neglected in the marine environments, has been now considered be a very active part of the marine trophic networks. And it is considered that just like the tip of the iceberg of marine fungal diversity is now discovered. So there is really thousands, if not millions of species to be described. So if you want to describe and name a fungal species, then this is the environment you want to look for to have, like to dedicate a fungal species to your beloved, for example. There is a very nice online resource if you want to inquire more about marine fungi. That is by Gareth Jones, Kevin Hyde, and collaborators. And you can just type www.marinefungi.org, and you can browse the species description. And you can also send to the committee that is reviewing the marine fungal diversity your publication and learn more about also fungal-like organisms that are inhabiting the sea. Where you can find them? Well, unfortunately, for now, the knowledge about distribution of marine fungi is very limited. So marine fungi for now are where marine mycologists are. Here is a review of marine planktonic marine fungi, and you can see I know a lot of people that are working on the topics, and so real high diversity in this zone, but a lot more to be explored. Where you can find that, what substrates? Here there is an image on the substrate where you can find, but I have to say these substrates are the most easier to study. If you're going to look more at other substrates, you will find marine fungi everywhere with the most amazing shapes that you can imagine. Why you study them? Well, at first, it was suggested that they were just measure the composers of woody and advanced substrates entering the marine environments. So they play pivotal roles, but now we know that they are integral part of trophic networks in the marine environment. The so-called microbial mycoloop. They are part of the mycoloop in the planktonic environment, and you will learn more about this in the next presentations. They are saprotroph. They are active part of the cycling of nutrients, and in particular in the carbon cycling. They are parasites, as you can see here. They are responsible for fish parasites, algae disease, and algal disease. They are symbionts. Every role that you can imagine. And this includes also they are degrading some materials that can enter and pollute the marine environment, such as microplastics. And why we want to study them? Because they are astonishing producer of bioactive compounds and enzymes. They are adapted to the marine environments. They produce anti-fouling enzymes, antibiotics, anti-cancer, really every compounds that you can imagine. And they are, most of them are very new and with a very innovative structure. This is just an example to finish my presentation. This is Dr. Huzefa Raja, and he was studying in 1944 the water of the port of Cagliari in Sardinia, Italy, because this water were really polluted. There were all the human sewage in there. It was a very nasty environment, but people were bathing in there and they did not get sick. So Brozu said, what? Again, what is happening there? What is the reason why people did not get typhus or enteritis? And he just isolated from the water of these ports, this fungus, marine strain of Cephalosporium acremonium, that later changed its name. And he started to see that the compounds produced by this fungus were astonishingly active against disease. And he asked for support from the Italian government to study these compounds, but he did not get any funding. So he sent this fungus to Sir Edward Abram that had funds and actually patented 3 antibiotics compounds, cephalosporin P and C and penicillin and he made a lot of money about that. So if you want to make money, that's another reason to study marine fungi. These are the funding agency of our project, and I really hope you will learn a lot more about aquatic and marine fungi today, and I'm here open for questions.
Thank you so much, Laura, for this lovely seminar talk about marine fungi. This is really such an interesting group of organisms, so diverse and so much potential here. So we already have a question from our attendees today, our colleagues, and we have a question that in the comment, I will read the comment because he says, I am interested in drug discovery from marine fungi. I am excited to be here. We are also very thrilled that we have so many people attending today. And how can be metagenomic guided isolation of marine fungi done? That's the question. Laura, do you have some answer?
Okay, there are 2 answers to these questions. One is drug discovery from marine fungi. You can isolate them. It depends on the substrates that you want to study. If you want to study algicolous fungi or fungi from the sediments or fungi from animals, you need different methods. I did not put the how, but there are a lot of resources that I can suggest if you want to isolate marine fungi, and then you can test them. Or if you want to make a shotgun sequencing on environmental samples, then you can look for example, PKS genes that are responsible for the production of many antibiotic compounds and anti-cancer compounds. So you can have from shotgun sequencing target genes that are responsible for the production of this kind of compounds, and then you can really study this diversity and see if this can lead you to drug discovery. But I'm a traditional taxonomy, so I like better to isolate fungi and to try them in laboratory because they can make like astonishing things, and you can really see the action against harmful bacteria.
Thank you, Laura. Maybe we have time for a quick question. I remember everyone attending today that you can please put your questions on the Q&A section because that's the only way we have it available today. Laura, if I can make you just a quick question. What would be your favorite species all time from marine fungi and why?
It was Corallospora marittima because it was like I was making my PhD. I started from scratch. And it was the species that I was shown. It was my first picture from my PhD. And it took me 2 years to get this obligate marine fungus. And I was so happy about that. And this is my favorite. I think of Corallospora as my relative.
Thank you. Maybe we have time for a very quick one. We have another question here. Grace says, thank you for the interesting talk. I was wondering what are the biggest threats to the diversity of marine fungi?
The thing is that we learn really few about marine fungi. So we will get the knowledge on their diversity when the environment, the marine environment is already changing. So, but there are really interesting papers about the roles of climate change and invasive species on the diversity of marine fungi. Climate change, because it changes the conditions, it can get rid of the host, but also invasive species, because invasive species brings basically less associated microbial diversity respect to those of the native species. So for this is, for example, another points that need to be taken into consideration if you study marine fungal diversity. But basically pollutants, increasing water temperatures, and all the human impact that you can imagine.
Thank you so much. We need to move forward to the next speaker, and I hand over to Monica to present.
Wonderful. Thank you, Isabel. It's my absolute pleasure now. We're moving from marine to groundwater, I hear, and it's my absolute pleasure to introduce Dr. Alice Retter from, she's based at the IGB Berlin in Germany, where she started working on the project FUNACTION that you heard about in the introduction, one of the hosts here today. Prior to that, she did her master's degree in biodiversity and systematics at the University of Gothenburg in Sweden, and then moved to Vienna to study for her PhD there at the University of Vienna. And that was in the group working on groundwater ecology. So really, Alice will be telling us a lot about what she found out during her PhD studies on these remarkable organisms. Alice, over to you.
Yes, thank you so much, Molly. For the nice introduction. I will share my screen. So, yeah, hi and welcome everyone. As you've already heard, I'm also part of FUNACTION with all these lovely people. And yeah, well, how should I put it? If you thought that the marine environment is not well understood and explored, yeah, let me top that with fungal diversity in groundwater, and let's see, yeah, what we find there, and maybe you hopefully know more after this presentation than before. So I thought I will present to you a little bit a picture of fungal diversity in groundwater with a little case study. And how it can look like, and also how does it look like compared to other freshwater habitats and terrestrial ecosystems. And in this little study, we explored a 300-kilometer river valley, which covered an elevation gradient of 1,800 meters in the Austrian Alps during spring and summer. So to give a little introduction into groundwater, it is a very temperature stable, light deprived ecosystem. It's poorly accessible. It is huge. It has the aquifers contain large amounts of volumes. It is containing 70% of the liquid freshwater on Earth. But due to its remote location, it received very little research up until now. Groundwater aquifers are also characterized by a scarcity of nutrients and therefore also very slow metabolic rates. Groundwater is of course part of the hydrological cycle, and it does not exist independently, and it is often more or less in a strong or weaker exchange with streams, lakes and wetlands, and also often sustains them. We are also very highly dependent on groundwater in terms of access to potable water, irrigation, industrial purposes, and so on, just to name a few. So when we talk about fungi, the main questions with groundwater is always, we cannot be certain how many species are purely transient, or if there are any native or autochthonous fungal communities, how the diversity looks like, if they are always just washed in, or if there are native communities there, and if they are very different to other ecosystems. Yep. So as Laura already mentioned a little bit, we all know the like fungal diversity is huge, but we all know the macroscopic fruiting bodies that we also like to eat from the forests and terrestrial habitats that are mainly composed of this of Dicaria, like Ascomycota and Basidiomycota, but many of the more ancient lineages are very abundant in freshwater ecosystems, and many more are to be described there, as most of the described species are Ascomycota and Basidiomycota, and only 1% of the described species are these more early diverging ancient lineages. So when we compare habitats, which is shown here, so we have samples from overlying soils, then underneath the groundwater and an adjacent river. And we can see that actually the fungal communities are very different from each other. And even the fungal communities within the groundwater are very different and very heterogeneous among each other than compared to the river and the overlying soil. So they all have their own microbiome. What we can also see, if we look a little bit closer into the communities and the diversity of fungi, that the groundwater is much more heterogeneous along the river valley than the fungal communities that we find in the river. Also, there are different phyla. There are differences in the taxonomic composition between these freshwater ecosystems. We can see that, for example, chytrids that are often parasiting phytoplankton are, of course, much more abundant in the river because obviously the groundwater is non-photic and they have no hosts there. So, but still we find some, for example, these red ones, Rocellomycetes, and also we find a lot of Olpidiomycetes, which are super ancient and super interesting lineages of fungi. And obviously, also not to such a surprise, seasonality is less pronounced in groundwater and is very pronounced in the river. And then we were also asking ourselves, what could be driving this composition? Is it very stochastic, or is there anything that dictates what communities we find in groundwater? And what we could see with our data is that hydrological characteristics, like how intensively the groundwater catchment is in contact with the surface through surface runoff or with through the exchange with adjacent surface water ecosystems, and also land use, like agricultural land use or urban areas. And also the river stretch, so the spatial distances, and also environmental characteristics. And what we can see is really that the hydrology is the main driver of groundwater fungal communities. And interestingly, we could also find patterns because we also sampled the groundwater fauna, like other invertebrates. And so I think it's very cute that also invertebrates such as nematodes to the right here, or snails or amphipods, also seem to kind of be an indicator for certain groundwater assemblages, fungal assemblages. What we also measured was fungal biomass, and we could see that the biomass was much larger in the river, obviously, than in groundwater. So same as diversity and biomass are manifold smaller in groundwater compared to the river, but still but still we can find them, and they are distinct own communities. And we also compared the ergosterol measurement and chalcofluor staining and counting. And what we can really see is that with chalcofluor we can also cover or catch the chytrids, especially in the river. Obviously, this is not possible with just measuring ergosterol, which is just produced by Dicaria. Then we also looked at putative growth modes and primary and lifestyles in groundwater, and comparing it to the river, and also which makes sense. Like dung saprotrophs, pollen saprotrophs, and algal parasites are much more prevalent in the river. But to our surprise, fungi that form filamentous mycelia and dimorphic yeasts are very, very prevalent in groundwater, especially like Trichoderma, Cladosporium, Epicoccum, and also many, and mostly the fungi in groundwater are that could be identified were soil saprotrophs, animal parasites, and also hyperparasites on other fungi. So yeah, most of them were actually soil saprotrophs washed in probably from the overlying soil, or just the shallow groundwater being really a very dynamic habitat probably. So what we know, to summarize everything, is that groundwater habitats are very closely linked to its surrounding terrestrial ecosystems in a very non-linear complex manner, much more complex than river systems and the terrestrial surrounding systems, because they can be connected vertically and horizontally, and this can often limit the exchange of fungi from the surface. Hydrological exchange that is dictated by then rainfall and seepage through the soil, and hydrological connectivity to surface waters, as well as the land use, are very strong drivers of fungal community composition in groundwater. And unlike prokaryotic communities, which have recently been found to be much more diverse than overlying freshwater ecosystems, the fungal communities are not as diverse in the groundwater compared to the river, which is probably linked to a limited input of carbon from the surface. And their biomass is altogether also much lower. But what we could find is that fungi from all taxonomic groups can be discovered from groundwater, and they harbor many early diverging lineages, such as Olpidiomycetes and Ocelomycetes, and many of them form filaments or yeast states, like these hyphal networks. And the chytrids that are usually co-occurring with phytoplankton bloom, including cyanobacteria, are obviously more abundant in the river systems, but what we found was that there are many soil chytrids, like this Rhizophleuctis rosea, that are find a good habitat in groundwater apparently. Yeah, and altogether we can say that groundwater is a unique ecosystem in terms of its microbiome, and it's its own very hidden world that just screams to be seen and more explored and not forgotten. Yeah, thank you so much for your attention, and if you have any questions.
Well, we do have questions, absolutely. Starting with the question, how can one learn to isolate and culture fungi? Are there any resources that explain this well? This anonymous attendee has tried several resources, but they all seem so vague. I feel what would be really good is if we could possibly post some of that in the chat throughout the webinar as well, because I feel all of our panelists have something to add to this. But if you've got any particular perspective, Alice, what did help you most?
Oh, I did this was high throughput sequencing.
Okay, good.
I have not isolated anything.
That's
I'm sorry.
That's okay.
Yeah,
We're not wanting to put you on the spot.
Yeah, I'm not the person to ask for isolation. I'm sorry.
Yeah, I feel there's plenty of folks who can probably put something in the chat to answer that question on our panel here. We've got a question. Did you find any trimycetes associated with animals? I'm sorry if I butchered that term.
I have not checked the fauna that we collected for any fungi actually, but we did sequence some, and I haven't looked at the data yet, so it would be super interesting. But no, I haven't looked. We just taxonomically described the major groups of fauna that occurred in these at these groundwater wells, and we didn't look further. But yeah, obviously, I'm always thinking they could be really, really interesting vectors for fungi. Also macrosporidians and all these parasitic, and then also hyperparasitic. I think it's could be a lot of weird stuff existing down there.
Cool. I feel in the interest of time, because we've got two more amazing speakers, we probably have to move on. But if our panelists could have a look at the Q&A, there's some very general questions of how do you measure fungal biomass in aquatic systems. If you have any wisdom to share, then keep checking the answers there that our panelists will put in. And I hand over to Isabel again. Thank you, Alice.
Thank you. And our next speaker today will be Serena Rasconi. Serena started her research on Lake Maggiore, working on bacteria and carbon cycle, and then she moved to Clermont-Ferrand in France, where she studied parasitic interactions among chytrid fungi and phytoplankton. Currently, she's a research scientist at INRAE Cartel, Alpine Center for Research on Lake Ecosystem and Food Webs at Thonon-les-Bains in France. And she's actively working on understanding how biotic interactions drive the functioning of our aquatic ecosystems, with a strong focus on algae and chytrid phytoplankton interactions. Serena, I hand over to you.
Thanks, Isabel. It's a great pleasure for me to be here today with you celebrating biodiversity. So thanks for the invitation. I will also have a short presentation going a bit into details into this group of aquatic fungi parasite of algae, which are chytrids that have been mentioned already a couple of times. And just a little hint about their role in the food web. So I would like to start with a short historical introduction to acknowledge a bit the scientists that started working on this organism. Chytrids have been described in the early 60s by Frederick Sparrow and Hilda Kanter that did a really nice job in observing and describing this organism. The books are really beautiful. So talking about biodiversity, I think this is something that we need to mention a bit also, how previously was really put effort in describing and also in details, really amazing details, the diversity of this organism. So, coming back to chytrids, chytrids, as I say, are fungi adapted to aquatic life. We are talking in this talk about parasite on algae, so we are back to open water, let's say. I'm working in lakes, so these examples are from lakes, but as Laura mentioned, now is known that, and Laura and Alice mentioned, now is known that chytrids are present also in the marine and river environment. But so coming back to chytrids, just a little overview on their life cycle. So they are quite a simple parasite with a life cycle completely usually in one single host. They're also called zoosporic because they have in their life cycle a dissemination phase, which is a flagellated spore which can swim and search for a new host. When the host is found, they can start developing into new sporangia and start the maturation phase, at the end of which, the zoospore are released in the water and searching for a new host for starting the cycle again. well, as has been mentioned, one of the method that is most used today is certainly molecular biology and metabarcoding, which helped a lot to highlight a bit the presence also of this organism in different environments were not well known. But what is always nice to do is to observe also this organism, and well, microscopy today is a bit more advanced than it was before. We can rely on epifluorescence, on different stain. So it's possible to discriminate easier now from fungi, from other organisms that are also on algae, but they're not, at least they're not fungi. If there are some parasitic relationship, these are not fungi. Or also it's possible to get a lot into details and even obtain some visualization of the zoospore into the sporangia. So it's getting really, the knowledge on this group is really growing, is getting a hot topic, and well, this parasite has been shown to be quite ubiquitous, present in different aquatic system, different lake, different algal species can be infected. Here are a few pictures also from colleagues in the Chytrid consortium. So yeah, as I say, there is a growing interest on this organism, and also given by the fact that this parasite can infect algae that can be interesting on an ecological point of view, for example, well, harmful blooming cyanobacteria, or even now we know also species of economic interest that can be highly relevant for the microalgal biotech industry, well, such as Hematococcus. So for this reason, we decided to formalize a bit the Chytrid consortium, which exists now since about 10 years, into a cost action. The cost action per aqua aim to share knowledge on this organism, and we organize different networking activities, and one of these activities well are the training school, and well, one of the first one was about detection method, where we showcased protocol based on dual staining on the algae in Lake Steklin. So, well, now that I introduced a bit this organism, and it's a bit more clear who they are and how to find them, I would like to get back to our lake and have just a little hint of what can be the importance of this organism in the food web and how they interact with other organisms. Well, as Isabel said in the introduction, I work mainly with trophic transfer and how the energy flows are going through the food web. And well, what we know, especially in the pelagic environment, is that mother sites, when it's come to feeding relationships, so is often that grazer and especially filtrators are limited in the size that the prey they can eat. And well, while small algae are easily accessible to filtrators, we know that large algae are not, and these resources can be lost by sinking once the life cycle of this algae is ended, because they cannot be recycled up to the trophic level. Well, thinking about chytrids and how to include them in this picture, we can see now that these parasites are able to feed on this large algae and basically making accessible the resources that are contained in these large cells via the production of spores that are very small and easily grazed by also the small filtrators. So just, yeah, this was just a short overview on this organism. Of course, there is much more to say, but yeah, this is just, I hope, yeah, first hint to make you a bit more curious about this nice parasite. And yeah, just to summarize, chytrids, as we say, well, they are fungi adapted to aquatic life. It is true, they are also saprotroph, but in pelagic system, they are mainly known as parasite of phytoplankton, on zooplankton, and other protists. And yeah, as I said, they can have an important role in the food web as overlooked trophic link. So thank you.
Thank you so much, Serena, for this super nice presentation. I guess most of the time when we hear the word parasites, we get goosebumps because majority of the time we feel they are bad, they are not important, or they will be harmful. And this is totally the opposite. So it's so nice to have this example of how important they are. And while we don't have a specific question for you here in the Q&A, and I remember everyone attending, if you have any question for Serena, please write it in the Q&A. What do you think would be the most important message to tell everyone about the importance of these parasitic fungi and that they should also be protected in our aquatic ecosystems?
Yeah, I think you mentioned already. I mean, parasitism, well, is a trophic relationship, and of course, it's extremely important to take this into account. Well, parasites are mainly studied because of their economical interest, because they're pathogen, because they create problems. This is a bit also the case, for example, within the cost action. So we help people that needs to produce biomass to solve the problem that can have with contamination and parasite, because it is true, parasites are harmful and can create problem when you want to target some specific production. But as ecologists, well, we have to keep in mind that parasites are important. They intervene in the energy flows. They have, as Laura said, fungi particularly have the capacity to produce metabolites that are outstanding and not always considered. So this can also be something that needs to be taken into account to broaden a bit our view. So yeah, again, I think the point is all about the diversity of the function that every organism can bring to the ecosystem.
Thank you, Serena. Just sharing with you that we have a comment from Haber and Simons that says, chytrid parasites also reflect unknown biodiversity that is also important to preserve. Some chytrid parasites are alone in ordinal level taxa levels in the phylogenies we produce. So yes, we need to take into account that all diversity is important, and that's what we are celebrating today. Thank you, Serena, so much, and I hand over to Moni to introduce our next speaker.
Wonderful. Thank you so much. Last, but definitely by no means least, we've got Dr. Huzefa Raja, who is based at the University of North Carolina in Greensboro. He is actually part of the Department of Chemistry and Biochemistry there. And really his research very much focuses on the potential that secondary metabolites from these freshwater fungi, the group that he will speak about, the ascomycetes, the potential that they might bring to us, bring to natural product chemistry and so on, to drug discovery. He did his PhD at the University of Illinois before moving to North Carolina, where he then also did his postdoc, both of those under the guidance of Dr. Carol Shearer. And to be quite honest, everything I know about aquatic fungi, I have learned from Huzefa. So I'm super excited to hand over to him now. Huzefa, over to you.
Thank you, Moni. Thank you very much. Right. Good morning to folks in the US. Good afternoon to folks in Europe, and good evening to folks in Asia and Australia. Thank you very much for this invitation. I'm very excited to be here on the International Day of Biodiversity. I'm very excited to share some of my research that we are doing here at the University of North Carolina, Greensboro. I wanted to mention that I am not a chemist by training. I'm a systematist, taxonomist by training. So everything I'm doing here at the UNCG with chemists is is in collaboration with the chemist, and some of the work that I'm going to show you has been done by chemists, right? So basically, the first major drug discovery from fungi was of penicillin. It was isolated by Alexander Fleming while he was on vacation, and he came back and he looked at his petri dish and he found that the bacteria that he was working on were not growing, or there was a zone of inhibition where a fungus was growing, right? And after that discovery, Ernest Chain and Howard Florey and Norman Heatley all worked together to basically get penicillin into drug discovery and discovered the first ever antibiotic from a fungus, right? The folks on the left, Fleming, Ernest Chain, and Howard Florey, got the Nobel Prize in medicine and physiology, but Norman Heatley, unfortunately, was not recognized for this discovery. So this set the stage for numerous fungal drugs, right? And Alexander Fleming mentions in his speech that it was not, he did not discover it, nature discovered it. And so these are natural products made by microorganisms, essentially, right? So after that discovery, numerous other fungal drugs have come into the market. Example, griseofolvin is an anti-fungal drug. It's a fungistatic drug, so it doesn't kill the fungus, but stops the fungus from growing. Penicillin, obviously, is the first antibiotic from a fungus. You have cyclosporin, which is an organ transplant drug from Tolipocladium. Lovastatin is a cardiovascular drug, and fungolimod is a new drug from Eiseria, which is an insect pathogenic fungus, and it is basically a drug for multiple sclerosis. So basically what I want to say is that these ascomycete fungi have all been very useful in producing natural products that can be used for drug discovery, and that is exactly what our lab is trying to do. Use ascomycete fungi, but found in freshwater habitats, to see what they produce in nature and isolate them and study their chemistry and see if we can spin that into a drug discovery endeavor. So here I want to show you on this slide two different pieces of wood that were incubated. And under the microscope, what you can see here are fungal colonies, right? This is wood from Australia, and here is wood from Illinois, where you can see there's all kinds of other things. These little tiny fruiting bodies are fungi, but you have a nematode, you have a dead insect, and a bunch of bacterial goop, right? So fungi are essentially growing on this wood, which is a long-lasting substrate in aquatic habitats, and are constantly competing with other eukaryotes and prokaryotes, right? In order to do so, because of this fungal competition and this ecology that ensues on these submerged substrates, we are using that chemical language of these fungal microbes and the compounds they make for drug discovery purposes, okay? So how do we do this, right? This is a very interdisciplinary research. So the first step is the mycology step, right? That's where I come in. My expertise is in isolation and identification of fungi from freshwater habitats. Our lab also works on fungi from terrestrial habitats. So the first thing is we go collect the substrates, bring them back to the lab, isolate the fungi in culture, and then identify them based on morphology. I'm also, like Laura, a trained mycologist in morphology. So we look at morphology, isolate the fungus in culture, and then we use molecular systematics methods to place the fungus in a phylogenetic perspective, right? On the right, you see a bunch of petri dishes. That's what my lab looks like with all these fungal cultures, right? The next step is where the chemists come in, and I work with the chemists very closely to do the first half of the project, right? So basically, I'm growing these fungal cultures in solid state fermentation states, essentially on rice, oatmeal, and sometimes even Cheerios. Basically, then the next step is extracting the fungus, right? Extracting is almost like how you would take tea and you put it in hot water, right? You're extracting all those compounds. So essentially that's what we do. Instead of water, we use organic chemicals, right? So the fungal samples are grown on rice or oatmeal, and then they're subjected to solvent extraction, right? So we have different partitions. We get sugars, we get fats, and then we get secondary metabolites. We are interested in the secondary metabolites. The sugars and the fats are kind of discarded for our purpose. And then the analysis continues with the secondary metabolites. Then we do a process called as chromatography, right? Chromatography essentially is, and when I say we, I mean the chemist, right? So chromatography essentially is a separation of these compounds which are in an extract. The extract has a mixture, and for drug discovery purposes, we want to isolate and purify these compounds, right? So chromatography helps with that. And that happens, basically, the separation of compounds happens on weight, polarity, and other chemical properties of the molecule. Once chromatography is done, then we are interested in finding the molecule mass, right? We're trying to identify the molecules. There are two ways of identifying the molecule. One is by mass spectrometry, a technique which is essentially an instrument that has a really good nose. It can sniff molecules, right? So it's essentially sniffing these molecules and telling, this is a molecule A, this is molecule B, right? And it's weighing the molecule by its atomic mass, right? So for example, we have this molecule here, C12H17O4, right? So this is all the calculation based on the atomic mass, and then a mass spectrometry gives you that peak right here, and then we know that this is based on this molecule structure, right? That's how the molecules are identified, right? And then we do something called NMR. And NMR is a method which is much more complex, but essentially it is when the molecule is new, how do you identify with mass spec? You have to actually try to put together the molecule piece by piece. And NMR, called nuclear magnetic resonance, helps in putting together the molecule, and you get the structure. Once you have a pure compound, then we do all kinds of antimicrobial activity assays, all kinds of cancer assays, what have you, against a biological target. So I want to tell you a quick story of a fungus that we found on submerged wood, right? Once this wood was collected, brought back to the laboratory, and incubated in boxes, I found this fungus, which is a disc-like fungus. It's a discomycete fungus, essentially. And what I found was these water droplets produced. The entire piece of wood was covered with these fruiting bodies, and they were producing these guttates, these exudates, right? And you can see these exudates on mushrooms while walking in the forest. You can also see, if you're working with fungal cultures, you often see these exudates or guttates in your cultures, right? These are not merely water droplets. And we were interested, and our lab has been interested in trying to figure out what is the chemical composition of these guttates, right? Essentially, in short, these guttates are chemical cocktails produced by fungi, right? And they produce them so that the fungus can then interact with its environment. If there is a bacterium, another fungus, or an insect in the vicinity, it is telling it, do not come near me. This is a chemical bomb, right? So what we did was, and this work was done by Manuel, who was a postdoc in our lab, is an excellent chemist. So he said, let's take these guttates with a pipetter, right? And we took that, and we got a really tiny amount, obviously, from several small fruiting bodies. And we got about 25 microliters, right? So it's a really tiny amount, less than an ml, right? We put that in a vial, and then we inject it into a chromatography system coupled with a mass spec. So we can see the separation of the compounds, right? And the weight of those compounds. So here's what we found, right? So you can see over here, let me get my marker here if I can, laser pointer. And so here you can see when we injected the guttates, what you want to see here on the x-axis is the time, and then you have the relative abundance here, and you can see several peaks, right? These peaks are not of interest because they are fatty compounds. We don't care for those fatty compounds. We want the small molecular weight compounds. So when the chromatogram and the mass spec was run, you can see all these different peaks. So this was the first peak. You can see at 5.23. Which is the retention time. It is the time when the compound shows up. And what we saw was this particular peak here at 464. So that's the molecular weight of the compound, right? That was the first compound that we saw. Then we saw that another compound came up here at 7 minutes, and that was the molecular weight of the compound, right? Interesting. So we found, and there were also 3 other compounds, right? So we found all this information. Essentially, what that meant was that this particular guttate, this liquid droplet, had 5 chemicals, secondary metabolites in it. So that's very interesting. And what we found was that these masses did not match with anything in the database. So essentially what this is saying is that these compounds are new to science. This fungus is producing these compounds, making these guttates, putting it out there in nature, and we don't know what they are made of, right? So this is interesting chemistry. So now we are going to work on extracting the fungus. So we are growing this fungus. We have a pure culture of the fungus, and then we will do more chemistry on it. What we want to see is that is the constitution of the guttate different from what is inside the fungus, right? And essentially, this is novel chemistry, folks. So then what we try to do is we try to send it to a collaborator who does antimicrobial assays. So Dr. Joseph Falkenham at Virginia Tech ran an assay on all of these compounds, right? Remember, it was a really small amount of guttate. So, but unfortunately, none of these compounds, none of these 5 compounds that we found had any activity, right, against antimicrobial targets. So we are going to work on doing further assays to try to figure out what these compounds are active against, right? So in our lab at UNCG, we have Thea Cameroon, a UNCG graduate student, currently working with freshwater fungal samples. So she's extracting these and isolating pure compounds and then testing them against antimicrobial targets at UNCG with Dr. Nadja Chek. We have an anti-malarial assay with folks, Dr. Dennis Kyle and Stephen Maher at University of Georgia at Athens. And Dr. Joanna Burdett is testing these compounds against cancer cells to see if they are active against cancer cell assays, right? All of these targets are important. We need new treatment modalities for all of these diseases. Antibiotics are a problem. There's antibiotic resistance. Malaria is a big issue in Africa and the tropical world. And obviously cancer, everybody has somebody in the family that has had cancer. And this is a very important target for us. And obviously this is all happening with Dr. Nicholas Oberlies as a lead at UNCG. And we are trying to test all our pure compounds from freshwater fungi against these different targets. So in conclusion, I just want to say that freshwater fungi are an understudied source of secondary metabolites. And what I have on the right here is a paper that we published, a review on the chemical diversity of these fungi. There's a QR code. If you're interested, you can take a picture and it'll take you to the paper, which is free. You can access it free of cost. And on the left side, I have another review on freshwater fungi, which was written for the Encyclopedia of Life Sciences. It is unfortunately behind a paywall. To access that, I have my email there. Please send me a message and I'll be happy to forward that paper to you. So totally, thus far, there are 280 compounds from freshwater fungi that have been described and isolated, and 199 of them appear in the review on the right? So finally, I would like to thank all the chemists at UNCG and all the collaborators that work with us. I would like to give a shout out to Moni and Isabel for inviting me to the session here, and to FUNACTION, MoSTFun, Global Species Center for Survival, and the Aquatic Fungal Species Conservation Group for this invitation. And I thank you all for your time. Thank you very much.
That's great. Wonderful. Thank you, Huzefa. We've got quite a few questions popping up in the chat. I can see that already some of our panelists are answering some of them via typing. For you, Huzefa, though, do the fungi make the compounds in the lab the same as in the natural environment? Do fungi release these compounds in freshwater and marine environments?
That's a good question, right? It's really hard to know because we don't, we have to bring the organisms back in the lab. So we really can't quite study them in the aquatic environment. So I'm sure they're different, right? Because once you bring the culture back to the lab, you don't have as much competition that you had on that piece of submerged wood. So that is definitely different. We do have an in situ technique where we can measure the compounds, but we haven't tried it with a piece of submerged wood, because essentially we would have all kinds of signals, because there's all kinds of bacteria and all kinds of fungal hyphae fruiting on the wood. So it would be really hard to figure that out. But I think the answer to that question is no, they're definitely going to be different, because the competition that you have in aquatic environments is not the same as the competition you have in the laboratory.
Excellent. And then there's a question that may be similar or different. I'm neither mycologist nor chemist, so I don't quite know. Don't the guttates that you talked about change with the growth conditions, including interactions?
Yes, yes. We have found that the guttates dry up over time, right? Because now they don't need to be made. Obviously, after the sample is sitting there, in fact, fungal communities on the wood come and go. So you have this fungus, and then after 2 months, that fungus is gone, right? So the guttates are gone too. So yeah, they do dry out, and perhaps the fungus. One hypothesis I have that this fungus is making it so that it's telling the other fungi, "Hey, let's all fruit together," and it's a communication compound. So I don't know what the target of this particular guttates are actually, and that is something that I'm really interested in finding.
Awesome, wonderful. Thank you so much, and thank you so much. Oh, there's another very quick question coming in. Has a plate contamination ever happened to produce metabolites of interest? That sounds
I have
exciting.
had that happen several times. And like Alexander Fleming, I never throw my plate away. So you never know. But yes, it has happened. And we found some interesting things sometimes. Yes. And I always want to go on vacation so that when I come back, some plates are contaminated and we find the next antibiotic.
Wonderful. Fantastic. Thank you so much, Huzefa. And thank you so much, everybody, for joining us today. I hope you found this whirlwind tour of the different wonders that really are there in terms of fungi in our waters as fascinating as I did. Every single time I attend one of these webinars, I learned so much, and most importantly, I think I learned that well, these are really groups that are so vastly understudied but hold so much promise and so much excitement, really, in terms of what we can discover from them. So it's really great to see all the latest developments, getting these various projects, FUNACTION, MoSTFun, and also the IUCN SSC Aquatic Fungi Specialist Group off the ground to really kind of highlight these organisms on an international stage, which is great. Thank you very much for attending. We will make this recording available, and we'll be in touch with where you can find it and where you can share it. I put links to all the different host organizations in the chat, as well as links to the social media. Please interact with us. Come check out these different projects, and keep an eye out for more webinars coming up that will discuss aquatic fungi and the wonderful and mind-blowing world of them, to be quite honest. Thank you very much.